Oh, hello, everybody. Welcome back again. We are starting the last chapter of the pure chemistry section of the course. So if you've found yourself struggling with the chemistry side of biology, the good news is that after this week, we move away from a discussion of pure chemistry and chemical pathways and chemical reactions, and we move into a look at more cellular processes where we're talking about cell division, genetics, inheritance, the role of the DNA molecule, synthesis of proteins and polypeptides. Some of that can be construed as being chemistry, and we will talk a little bit about chemistry there, but in terms of being purely focused on the chemistry of cells, we're done with that as soon as we get to the end of this chapter at the end of this week. So perhaps a little bit of relief for those of you who've been struggling with the chemical side of biology. But we still need to get through this chapter. And I'll be honest with you, in case you have not already picked up on this, I think I've mentioned this before, but I am a plant scientist. That is my background. That's what I did my research in. That's what I did my PhD in. And so I really enjoy talking about plants, and I really enjoy this section talking about photosynthesis because I get to talk about some of my professional interests. So one of the problems that I really have with talking about photosynthesis is keeping it simple and keeping it brief. We'll see how I get on this time. The section is going to be broken up into 4 chunks. Broken up by concept checks. The second one is going to be relatively long, probably because we're talking about the actual photosynthesis process or the light capturing part of photosynthesis. So that one is likely to be a little bit longer than the others. The others will be probably reasonably short, but again, it's going to be in the format that I introduced with the cell respiration chapter, where I will stop the recording and then restart the recording at each concept check and answer the concept check question with the beginning of the new recording. That seems to be reasonably popular, and it also does allow for those of you who like to do the whole recording in one session to continue straight on through. There is a button at the bottom or an arrow at the bottom that you can click on to take yourself forwards into the next recording as you wish, so that you can move through it at your own pace if you've got everything that you need to get out of each recording. So that being said, let's get started. So why do we want to talk about photosynthesis? Well, apart from the fact that it is a partner process with cell respiration, what we also need to remember is that this is how carbon gets into all organisms. This is how carbon enters the biosphere. It is organisms that carry out photosynthesis, whether they be plants or algae, cyanobacteria, anything that is green, has chlorophyll, carries out photosynthesis, is contributing to the influx of carbon into the biosphere. And the vast majority of carbon comes in via this process, which is the reason why we really want to talk about this. This is where all the carbon that's found in you and I and every other organism on the planet, and we might as well throw in there as well all the carbon-containing fossil fuels. So things like crude oil, natural gas, coal, peat, that we might use as a source of energy by burning these fossil fuels. Those are all also the product of photosynthesis because they are the dead fossilized remains of organisms that have lived in the past. In fact, if we want to look at the largest reservoir of carbon on the planet, we look no further than life as we know it right now and life that has existed in the past in the form of all of those fossil fuels. So if you really want to bring this firmly home, just bear in mind that every last carbon atom in your body has come through photosynthesis. and specifically pass through a single enzyme that we'll encounter when we talk about the Calvin cycle, the second half of photosynthesis. And that enzyme is abbreviated rubisco. I'll give you the full name of it twice during the course of that section of the presentation, and you'll understand why we like to abbreviate it rubisco. But every carbon atom that has entered into the biosphere has come through that enzyme. So we do really need to talk about that side of photosynthesis, even if we don't talk about anything else. But we do need to talk about photosynthesis as a process, because this is what is providing the ultimate source of energy that keeps all life functioning on Earth. Yes, we get the ATP that we utilize through cell respiration, but unless photosynthesis occurs, there would be no organic molecules for respiration to process to provide the energy to make the ATP. So we can't divorce one from the other. We really do need to consider the two together. So the first thing to recognize is that plants and any other organisms that carry out the reduction of carbon dioxide to organic molecules are correctly called autotrophs, meaning they create their own food. Now, the reason why I say anything that can reduce carbon dioxide to food molecules is because, as you'll see shortly, there is a partner process to photosynthesis, which we refer to as chemosynthesis. And it has a very important role historically in our understanding of how photosynthesis actually takes place. But not a lot of organisms do it. But there are a small number of organisms that do it, and it probably will come as no surprise to you that it is bacteria, prokaryotic cells, that carry out chemosynthesis. There are no eukaryotes that actually carry out chemosynthesis. But it was coming to terms with one form of chemosynthesis that actually allowed us to fully understand what photosynthesis is all about and how it occurs chemically. But more of that shortly. So autotrophs have this ability to take carbon dioxide from the atmosphere and reduce it to organic molecules. We say glucose only because it allows us to chemically directly tie cell respiration and photosynthesis together. Remember, glucose is the primary fuel for all organisms, and we write that as the primary organic reactant in cell respiration. The byproduct is carbon dioxide and water. Those become the reactants in photosynthesis. And of course, photosynthesis takes the carbon dioxide and reduces it to organic molecules. And in case you hadn't figured this out yet, that reduction requires electrons, and those electrons are sourced out of the water molecule. So if we think in terms of the relationship between cell respiration and photosynthesis, we see this chemical cycling going on. But now we need to actually think in terms of the electrons. We know that the electrons from glucose are the key for cell respiration because they become the energy source via the electron transport chain for creating the hydrogen ion gradient that actually directly drives the ATP synthase to make ATP. And those electrons energetically run downhill. They go from the high free energy environment of NADH to the low free energy environment of oxygen and water. So we see a decrease in the overall free energy of the electrons. They go downhill. So it stands to reason that if we are reversing cell respiration, if we are going to take carbon dioxide and water and use them as the raw materials to make ultimately glucose, according to the basic reaction, which, as you'll see, is not entirely correct, but it nonetheless serves a purpose, then if the electrons are running downhill in cell respiration, the reverse requires the electrons to be moved uphill. And of course, if you're going uphill, you're doing work, that means you need an input of energy. And as we have talked about before, using an energy molecule to make that energy molecule, in this case, using ATP to make ATP, is a no-win situation. It's a zero-sum game, meaning eventually you're going to run out of ATP. if that's what you try to do. So the only alternative is to come up with an external source of energy to generate the ATP. And ATP is a part of photosynthesis. So is another molecule that's going to sound very familiar, NADPH. Sounds very much like NADH that we've seen in cell respiration. And it serves exactly the same purpose as NADH serves in cell respiration. That is, it allows for the transfer of electrons from one location to another. And remember, again, photosynthesis is all about electrons because we are ultimately going to want to reduce carbon dioxide to organic molecules to sugars. We think primarily glucose, but as I'll demonstrate, that's not necessarily how we should think, but it serves the purpose of creating that connection back to cell respiration. But we have reduction going on. And of course, as we learned in cell respiration, if you're doing a reduction, you must be doing an oxidation. You can't have one without the other. So that means we must be sourcing the electrons from somewhere, and the water turns out to be the source of those electrons. So we can start thinking in terms of chemical reactions, and we can start thinking in terms of what goes on in photosynthesis. So this just diagrams the types of organisms that hopefully you're all familiar with that carry out photosynthesis. But these are the guys that are going to serve our purpose in terms of helping us chemically understand what's really going on in photosynthesis, because these are the organisms that carry out chemosynthesis. So let me just make sure that the pen's in black. And these carry out chemosynthesis. So the purple sulfur bacteria, and there are also some green sulfur bacteria, carry out this process of chemosynthesis. And it's the purple sulfur bacteria that in the 1930s, being studied by an American biologist named Van Nile, who figured out that what the purple sulfur bacteria are actually doing is a redox reaction involving not water, but something that we've talked about in cell respiration, H2S, hydrogen sulfide gas, which is an alternative byproduct to water in organisms that carry out alternative forms of respiration that we refer to as anaerobic respiration, where oxygen is not the final electron acceptor. Well, it turns out that H2S, like water, can also function as a source of electrons. Not only is water formed in cell respiration and H2S formed in anaerobic cell respiration as the result of electrons passing through the electron transport chain in the mitochondria, both H2S and H2O can serve as donors of electrons in H2O in photosynthesis and H2S in chemosynthesis. And it was Van Nile's examination of the chemistry of purple sulfur bacteria and figuring out that it was actually a series of redox reactions that finally put together the pieces of the puzzle that solved what is truly going on in photosynthesis and actually resulted in a rewrite of the chemical reaction for photosynthesis, which you will need to know. You'll need to know the rewritten equation because it is the correct chemical equation for photosynthesis. And you may have learnt the quote unquote old equation. And again, that's not entirely inaccurate. It is a balanced equation, and chemically it does show photosynthesis as a redox reaction, but it doesn't actually demonstrate or take into account what we now know is the functional chemistry of photosynthesis. So if we look inside plants for the location of photosynthesis, we know that it takes place in leaves. We know that it takes place in cells within the leaves called mesophyll cells. So if you see the term mesophyll then you're basically talking about the cells that carry out photosynthesis. It's not going to fit. Let's erase that. Just put it in here, we got space. So whenever you see the term mesophyll, you're dealing with cells that contain chloroplasts and carry out photosynthesis. And there are two basic types of cells in leaves that carry out photosynthesis. So you can see the two different types here. Let me just move this box aside for a minute. So these are called palisade mesophyll cells, and these are called spongy mesophyll cells. And the palisade mesophyll cells are called palisade cells because they look a little bit like the wall of an old log fort where the logs are stood on end. That's called a palisade. And so when biologists saw these inside cells, someone decided to call these palisade mesophyll cells. And then the spongy mesophyll cells are called spongy because they contain a lot of air spaces around them. And those air spaces are actually important because that's where the gases accumulate, the carbon dioxide that's coming in for photosynthesis and the oxygen that's leaving as a result of photosynthesis, as the byproduct of photosynthesis. And those gases move in and out of the leaf through those openings called stomata that you can see there. Now, in a later section in this presentation, we'll talk about some variations on photosynthesis, and those stomata become important because it turns out that these are also a major point of water loss from the leaf. And so for a plant to move water through its the body of the plant, there has to be a certain level of water loss through the stomata to allow water to evaporate off the surface of the cells to create that sucking force that we talked about back in chapter 3 that allows water to move up plants and to move up trees. It all originates here in the leaves as water is lost through the stomata. But if too much water is lost, the plant becomes severely dehydrated. So the stomata and their opening and closing are regulated by a pair of cells called guard cells. And it's this requirement to regulate the amount of water loss that's going to become critical in understanding some of the variation in photosynthesis mechanisms and ways plants have adapted to different environments by being able to regulate water loss by regulating the stomata and still be able to carry out photosynthesis. So more of that to come a little bit later. When we take a look inside the chloroplast, and this should not be a foreign diagram to you, we've looked at this diagram before. We know that the inner membrane system is highly folded into these little discs called the thylakoids. There is a space inside called the thylakoid lumen, or the thylakoid space. Remember, all of the spaces are connected together to form one large space inside these stacked discs, the stack being referred to as a granum, the plural being grana. And each granum is connected to adjacent grana by these connecting membranes. So we have a large internal compartment formed by the thylakoid spaces, separated by the second compartment of the chloroplast, the stroma. And so the separating membrane is the thylakoid membrane. That sounds a little bit like the internal makeup of a mitochondria, and that's good because it's going to turn out that these two compartments and the separating membrane are important, just as they are in mitochondria for the generation of ATP, so they are here in photosynthesis for the generation of ATP. In fact, ATP is made in chloroplasts using exactly the same mechanism as we've seen in mitochondria. More to come shortly. So this should not be a completely foreign idea, the location of, or the structural features of a chloroplast. So let's start putting the bits and pieces together. If we have a lot of membrane, we know that that must be important. Having the stacked membranes must also be important because we know cells don't create structures without some benefit. There's got to be some purpose. And so it's the stacked membranes of the thylakoids that are responsible for capturing light energy. The actual complexes are embedded in the thylakoid membranes, along with the components of an electron transport chain and an ATP synthase. So again, if this sounds a little bit like a mitochondrion to some degree, that's good, because again, we're going to be looking at a very similar process. The big difference here, of course, is the membranes are involved in capturing light energy as the driving force behind photosynthesis. Then we have the stroma, and we have enzymes in the stroma that are responsible for reducing carbon dioxide to sugars. So that's going to take place in the stroma using the captured light energy in the form of ATP as a source of chemical energy and electrons from the NADPH, which I've mentioned already, but we'll see its role again. Those sources of energy are going to be utilized by the enzymes in the stroma that form the Calvin cycle that to reduce carbon dioxide to sugars. We again primarily think in terms of glucose, but as you're going to see, glucose is probably not the best molecule to think of in terms of understanding the Calvin cycle, but more of that to come later. So when we start thinking in terms of cell respiration and we think in terms of photosynthesis, let me move this over here so that it's out of the way. In fact, I'm going to move it all the way to the top so I can use all of this empty space. So when we think in terms of respiration, so respiration is C6H12O6 plus 6O2 gives us 6 CO2 plus 6 H2O. And we know that the glucose is oxidized yeah, let me just put it up here. So we know that the glucose is oxidized to carbon dioxide, the water is reduced to H2O. If you've studied photosynthesis before, then you probably learnt the following equation. So I'm going to put it in quotes because it turns out that this is not the correct equation. But you probably learnt it as 6O2 plus 6CO2. Not sure what I was thinking there. It's C6H12O6 plus 6O2. And you probably learnt that the carbon dioxide was reduced, if these terms were used, to glucose, and the water was oxidized to oxygen. And as far as that equation goes, I mean, it's chemically balanced, and it does indicate that cell respiration and photosynthesis are both redox reactions. Fair enough. The problem is that if you provide carbon dioxide containing an alternate isotope of oxygen, because it turns out that oxygen comes in two isotopes, oxygen-16, which is the most common form, and oxygen-18, which is a slightly heavier form. It's got two extra neutrons in the nucleus. If you provide carbon dioxide that contains oxygen-18, that oxygen-18 does not come out as the oxygen gas. The only time the oxygen 18 comes out as oxygen gas is if you provide the oxygen 18 in water, that it's the oxygen, that the oxygen 18 is the oxygen in water. And what that tells us is that the isotope, and I'm going to put this in red, I think, because it's kind of important. The isotope results indicate that water is the sole source of the O2. And in this case, that's not going to work because over here, you've only got 6 oxygens, but here you have 12 oxygen atoms. And so if the water is the sole source of the oxygen, there's simply not enough water molecules as reactants to give rise to the oxygens that make up the oxygen gas. Now, this had already been hinted at by the work of Van Nile. So remember I said that he was working with the purple sulfur bacteria. So the purple sulfur bacteria with their chemosynthesis, and I think I'll write the equation in purple, seeing as how they're purple. Their equation, Van Nile figured out, went like this. So what Van Nile was able to show is that, yes, the carbon dioxide is reduced to glucose. So there's the reduction. The H2S is oxidized as the source of electrons to pure sulfur. I put an H in there, that's not correct. It should be S, it should be sulfur. So Van Nile had already discovered that the purple sulfur bacteria are actually using H2S as their source of electrons, and you get pure sulfur from that. If you go back and look at the picture of the purple sulfur bacteria, all those yellow blobs sitting inside each of the bacterium, those are blobs of sulfur. Those are the byproduct of the oxidation of H2S. And what Van Nile concluded from this is that, in fact, if this is a parallel to photosynthesis, and that the H2S is serving the role in what the purple sulfur bacteria are doing, as water is serving in photosynthesis, then the oxygen that's being produced by photosynthesis ought to be coming from water. And this chemical equation is incorrect. The problem was that Van Nile couldn't demonstrate it. But in using the oxygen 18 isotope, people could chemically show that what Van Nile had been thinking was absolutely correct. The way that you get around this problem of not having enough water molecules is simply to double the number of water molecules. So if we go to the next picture, the next slide, get it to change. That's what it has actually been been shown. So again, if we put in the purple sulfur bacteria, you see exactly what Van Nile had seen and why he considered that this would be a better equation for photosynthesis. It doesn't take away the redox reaction nature of photosynthesis. It simply now chemically accounts for where, and perhaps I want to change this back to red again. So let me just apply the eraser there. It simply takes into account the assumption that Van Nile had made that oxygen, that the water was the sole source of oxygen. He could not chemically demonstrate it, but we can now, using the isotopes of oxygen, that the water is in fact the sole source of oxygen. But it creates another problem. If you put in the 12 waters to give you enough oxygens to make the 6 O2s, you now have too many hydrogens, because you only need 12 hydrogens to do the reduction of carbon dioxide to glucose, because glucose is H12, not H24. But you've got 24 hydrogens sitting over here as a result now of having 12 water molecules. So 12 times 2, there's your 24 hydrogens. So the solution is, again, to come back to what Van Nile had discovered about the purple sulfur bacteria, and to realize that the hydrogens go to reducing oxygen to water, or some of them go to reducing oxygen to water. And so now, when we write the equation for photosynthesis, not only is water a reactant as a source of electrons, it also becomes a product as a source or a destination for some of these extra hydrogens that we've got. And so the only way that we can account for it is to do what Van Nile did. He just, bear in mind that he's still just dealing with the H2S. So as far as he's concerned, H2S, the S becomes the sulfur. These hydrogens, some of them are used to reduce carbon dioxide to glucose. The rest, well, they create the water molecules. So for him, it was a no-brainer. It was the solution to his equation. For us, it was a little bit more difficult to correlate the idea of water as a reactant and a product. But that's the only way we can make the equation work, given that we know that the oxygen is coming totally from water and from nowhere else. But we now have to account for those extra hydrogens. And so we take some of the oxygens from glucose, because we only need 6 oxygen, sorry, some of the oxygens in carbon dioxide, because we only need 6 oxygens for glucose, but we've got 12 over here, because we've got 6 times 2, that's 12, but we only need 6 for glucose. So we've got 6 extra O2s, or 6 O's, plus 12H, and that's what we've got in excess, and that's what becomes our water molecules. So that's the reason why this is now considered to be the correct chemical equation for photosynthesis. So as much as you may have learnt the equation that I wrote on the previous slide, this is the equation that you now need to know, because this is the correct chemical equation for photosynthesis. So make sure that this is the one that you understand, and that you understand why it is the correct equation. So once we start recognizing that we are extracting electrons out of water as the source of reducing power for reducing carbon dioxide, then we start thinking in terms of photosynthesis as a redox reaction, just as we saw with cell respiration. In cell respiration, remember, the electrons from glucose are running downhill to oxygen. because oxygen is extremely electronegative, low free energy. Glucose, not so electronegative, high free energy. Now we want to move the electrons in the opposite direction. We're going from water, low in free energy, high in electronegativity because of the presence of the oxygen, and we want to move those electrons uphill to glucose. So we do need a source of energy to push the electrons back uphill again. And so that becomes part of our understanding of photosynthesis. There are two basic steps, what are referred to as the light reactions, the way in which we capture light energy and use that to generate ATP as a source of chemical energy and to also promote the electrons energetically to move them uphill from water, low free energy, to NADPH, a molecule of high free energy, just as NADH is a molecule of high free energy in the mitochondria. Those electrons then will be used to reduce carbon dioxide to sugar in the Calvin cycle, utilizing the chemical energy from ATP and the electrons from NADPH. In the process, the ATP is hydrolyzed to ADP and PI, which then cycles back to the light reactions to be reconstituted as ATP. And the NADP plus that results from the extraction of the electrons, an oxidation reaction, so that we can reduce the carbon dioxide. The NADP plus is also cycled back to the light reaction so that it can be reduced again to NADPH using electrons ultimately sourced from water. So these are the two processes that are going on. The light reactions occurring on the thylakoid membranes and in the thylakoid membranes, generating the ATP and NADPH, which then cycles into the stroma to be utilized as the chemical energy and the reducing power for reducing carbon dioxide to sugars. We will come back and revisit this diagram at the very end of the presentation and fill in the blanks for the light reactions in the Calvin cycle, because there is a lot more detail to be filled in here yet. But that we can only do by talking about the light reactions in more detail and the Calvin cycle in more detail in the upcoming sections of the presentation. So, time for our first break. We've been going for about 40 minutes at this point, so it's a good time to stop and just take a quick break. Here are our concept check questions. Remember, if you're going to continue on with the presentation, I'll answer these at the beginning of the next recording, or you can walk away, take a break, process what's been discussed in this section, and come back and pick it up at your leisure and get, after you've thought about these concept check questions, if that is your desire. So here are the questions. Explain how oxygen isotopes and sulfur bacteria help figure out the chemistry of photosynthesis. And the energy for the reduction of CO2 is provided by NADPH and ATP from the light reactions. If a classmate insists that the light reactions don't require the Calvin cycle, how would you respond? An interesting question. I'll have the answers for those when we pick up the recording again in the next section. See you there.